Hi everyone, in Module 4, we're going to be talking specifically about the physiology of the heart, and the function of any pump is to develop this pressure and to move a particular volume of fluid in a specific direction at an acceptable speed. So this is exactly what the heart is going to do. And in this specific learning outcome, which is learning outcome number 1, we're going to be talking about the nodal cells. Therefore, we can say that the heart will work in these cycles of contraction and relaxation. If we're referring to contraction, it is also called systole, and relaxation will be called diastole. And the pressure within each chamber will alternately rise and fall during these periods of either contraction and relaxation. Now, the atrioventricular valve and the semilunar valves, they will definitely help to ensure that there is a one-way flow of blood despite these pressure variations. And blood will flow out of an atrium only as long as the atrioventricular valve is open and the atrial pressure will exceed the ventricular pressure. In the same way, the blood will flow from the ventricle into an arterial trunk, either the pulmonary trunk or the aorta, only as long as the semilunar valve is open and the ventricular pressure exceeds the arterial pressure. And the proper functioning of the heart will therefore depend on the proper timing of this atrial and ventricular contraction. So there's going to be a very elaborate pacemaking, something that will set the tone, the pace, and also an elaborate conduction system that normally will provide the required timing for these events to take place. And this is exactly what we're going to see in this module. There are two main types of cardiomyocytes or cells that belong to the cardiac tissue. These are the nodal cells. They can also be called conductive cells and the contractive cells. The nodal cells are what we call the pacemakers. So these are the ones that will set the tone. And the contractive cells are all other cardiomyocytes. So on this specific learning outcome, we will be covering just the nodal cells, and on the next learning outcome, we will talk about the contractive cells. Now, unlike the skeletal muscles, the cardiac muscle tissue, they will contract on its own. And this is even in the absence of neural or hormonal stimulation. And this inherent ability to generate and conduct these impulses is called autorhythmicity. And we have talked a little bit already about this, but the autorhythmicity just means that the cardiac cells, they are able to start to contract on their own without the influence of any neural or hormonal stimuli. But it is true that these neural or hormonal stimuli, they are able to alter the basic rhythm of contraction. However, even if a heart is removed from, for example, a heart transplant, it will continue to beat unless there are steps that are taken to prevent it from beating. In fact, if you click on this image over here, hopefully you'll be able to see that these cells that were removed from a rat heart and placed for culture in a petri dish, they are still beating, even when they are removed from the heart itself. Let's see how this autorhythmicity occurs in those nodal cells. We can say that these nodal cells, which can also be called pacemaker cells, where they are setting the pace for muscle contraction, it will spontaneously depolarize, which basically means that they will spontaneously send an electrical signal or action potential, and this will cause these cells to contract. And their contraction actually occurs at about 70 to 100 times per minute. Now, there are two locations where these nodal cells are present. one at the top of the right atrium, and this group of nodal cells are called SA node. The second group of nodal cells are called the AV node, and they are located near the coronary sinus of the interatrial septum at the bottom of the right atrium. And when both groups of cells they send this electrical signal, they will spread it out to the heart muscle tissue in the following way. From the sinoatrial node or SA node, right over here, notice how these electrical signals they go down to the right atrium and also across over to the left atrium through what we call the internodal pathway as seen on this image. These green arrows are going to be the ones that are representing the internodal pathways. And as you can see, it spreads through the myocardium of the atria that then tells the heart muscle to contract. And this squeezes and then pushes the blood from the atria down into the ventricles on both sides of the heart. Therefore, the blood from the right atrium will go into the right ventricle, and the blood from the left atrium will go into the left ventricle. Now, this electrical signal wants to spread down to the ventricles, but it actually can't because of this fibrous tissue between the atria and the ventricles, which will form the AV valves. So basically, this fibrous tissue prevents this electrical signal from going directly from the sinoatrial node into the atrioventricular node. But we know that this electrical signal will continue because they need to reach the ventricles to be able to contract the ventricles. So this electrical signal will be able to bottleneck itself through the other group of specialized myocardium cells, which are the AV node. And as the action potentials make their way from the SA node to the AV node, there's going to be a pause in the muscle contraction of 0.1 seconds, meaning that there is a break between the atrial contraction and the ventricular contraction of 0.1 seconds. And this timing is going to be important because it gives the heart enough time to fill up the ventricles with the blood after the atria contracts. And after this 0.1 second, the action potential will be able to spread down the AV node, and we can see from this image that it goes down into what we call the bundle of His, which is located in the interventricular septum. And this bundle of His will then branch out into the left and right bundle branches, and then they will send electrical signals through the ventricular heart muscles, and as it reaches the apex of the heart, this action potential then tells the heart muscle to contract. And with this ventricular contraction, the blood is going to be able to be ejected from the heart. And if this is exiting through the right ventricle, it will go to the lungs. And if it's exiting through the left ventricle, it will go to the systemic circuit and then spread throughout the entire body. Now, one thing that I want you to notice is that once the left bundle branch and the right bundle branch reaches the apex, they become the Purkinje fibers, which will be located at the apex of the heart. And then this is what will send the signal for the muscle cells in this area to start to contract. In addition, we can see that there's a small branch that will travel through the moderator band and also contribute to the contraction of the ventricle. Now, why does it travel all the way to the apex of the heart before it contracts? The reason for this is that you want to make sure that you expel the most amount of blood possible. So think of it this way. If you were squeezing a tube of toothpaste in the middle, which I'm sure is one of the pet peeves of a lot of couples, right? When the partner squeezes the tube of toothpaste in the middle instead of squeezing it from the bottom. So if you squeeze it from the middle, what happens is that you get toothpaste coming out through the hole and toothpaste staying inside of the tube. But if you were to squeeze the toothpaste tube from the bottom, then you would get all the toothpaste to be able to come out through the hole. So this is the same concept. You want to make sure that the heart is being squeezed, or that the muscles in the heart tissue are contracting from the bottom to be able to expel and push out the blood that's located in the ventricles. So how do the SA node and the AV node send their electrical signals? We have already established that the SA node spontaneously depolarizes, right? They just basically stimulate this action potential all by itself. But how does this happen? Well, first, let's look at the cardiomyocytes from the SA node, represented here in blue, and a regular cardiomyocyte represented in pink. Even though they are different, they do communicate with each other through these gap junctions that we talked about earlier, as we can see represented here on this image. So they are able to basically talk to each other and communicate with each other. And the first thing that I want you to know is that all the cells in our body, they have a very special pump that's called the sodium-potassium ATPase pump. And what it does, like the name says, is that it uses ATP, which is the energy currency of the body, to throw 3 sodium ions out of the cell and 2 potassium ions into the cell, as we can see from this image over here. Now think about this. If the cell is throwing 3 positive sodium ions outside of the cell and sends 2 positive potassium ions to the inside of the cell, where do we have a net positive charge? Well, the net positive charge is going to be outside, right? Do we not have 3 positive sodium ions compared to 2 positive potassium ions inside of the cell? Therefore, this means that the net negative charge is going to be inside. And if you were to compare those charge differences, what you would find is that the net difference is a negative 5 millivolts inside of the cell, as we can see represented here on this image. However, this is not all that's happening. What else is happening? Let's see. We are accumulating all this extra sodium outside of the cell, and we are accumulating a whole bunch of potassium inside of the cell from this active sodium potassium ATPase pump. Now, the cell membrane is actually quite leaky for potassium, meaning that the potassium ions will be able to leak out of the cell very slowly. And by doing this, it makes it even more negative inside of the cell, right? Don't you have more positive ions going out of the cell? And this will make the charge of the cell go from minus 5 millivolts to around minus 60 millivolts, as we can see from this image, which is the charge difference inside compared to the outside. As we move along, we also have channels that are going to be leaky to sodium and calcium ions. This means that the sodium that is outside of the cell is wanting to go inside of the cell. With regards to the calcium ions, these are going to be predominantly found outside of the cell. Therefore, it will also want to leak inside of the cell. And as we can see from this graph, there is an increase in the membrane potential from minus 60 millivolts up to around minus 40 millivolts. So what is really happening here? What this means is the following. The action potential process is going to start its baseline at around minus 60 millivolts from those leaky potassium channels. But because of this leaky calcium and sodium, it is always drifting up slowly towards a positive membrane potential. And once it reaches minus 40 millivolts, this is going to be the threshold for another group of channels. They're going to be a different type of calcium channel, which is going to be a faster type of channel. And this means that more calcium will enter. And when it does, it will bring the membrane potential suddenly to a positive 20 millivolts, which is what causes the depolarization of the membrane, and this will lead to an action potential. So the signal is sent for contraction. And once it reaches a positive 20 millivolts, another potassium channel opens up. And this allows potassium to exit the cell, and this makes, again, the cell less positive, as we can see over here. So once we have the rapid influx of calcium ions entering, this will cause the depolarization of the membrane. Once we have the outflux of potassium exiting the cell, this will cause the repolarization of the cell. Therefore, the cell membrane alternates between depolarization and repolarization. And because these nodal cells are connected to the contractile cells, this information will be able to be sent to the contractile cells for them to contract the atria and then contract the ventricle. And we will see this in the next learning outcome. This slide is more of a review of what we have talked about in regards to what's happening at the sinoatrial node and the atrioventricular node. I truly don't think that I need to repeat myself here, so if you guys have any questions, just let me know. Send me an email.